Welcome to Last Minute Lecture. This free chapter overview is designed to help students review and understand key concepts. These summaries supplement, not replace, the original textbook and may not be redistributed or resold. For complete coverage, always consult the official text.
I want you to picture a coastline. Like maybe you're standing off the coast of New Jersey or, somewhere down in the Gulf of Mexico.
It looks absolutely beautiful from the shore.
Right. The waves are crashing, the sun is shining, but under the surface, there is this massive dead zone, a vast suffocated region where, almost every plant and animal living below the waves has completely died off.
It's literally a biological wasteland down there.
Exactly. And to understand how an entire stretch of the ocean just, perishes like that, well, we are embarking on a very specific mission today. If you are a college student staring down a microbiology exam on marine and freshwater ecosystems, consider this your last-minute lecture.
We've got a lot of ground to cover.
We really do. We are fast-tracking a deep dive into Chapter 29 of Prescott's Microbiology, taking the dense material you need to know and translating it into clear sequential knowledge. Yeah. So let's start with that dead zone. How does a massive stretch of the ocean just run out of air?
It sounds like a paradox, but it actually starts with an overabundance of life.
Oh, really?
Yeah. The mechanism actually begins on land. We use massive amounts of nitrogen and phosphorus in agricultural fertilizers, right? So when it rains, that nutrient-rich runoff washes into the rivers and eventually hits the ocean.
Okay, so it all flows downstream.
Right. Combine that sudden influx of nutrients with warmer water temperatures, and you trigger this explosive, massive growth of phytoplankton. They multiply endlessly until the nutrients run out, and then they inevitably die and sink to the bottom.
Okay, so a massive cloud of dead phytoplankton hits the ocean floor. That seems like it would just be, a buffet for whatever is living down in the mud.
Oh, it is a buffet, but with disastrous consequences. Yeah. Because the seafloor is packed with heterotrophic microbes.
And those are organisms that consume organic carbon for energy, right? Like we do.
Exactly like we do. So suddenly, millions of tons of food literally rain down on them. Because the food supply is nearly infinite now, these heterotrophs multiply at an astonishing rate. But here is the critical mechanism. As they gorge on the dead phytoplankton, they respire. They consume dissolved oxygen.
Right, they have to breathe.
And the flux rate of oxygen moving through water is roughly 10,000 times slower than it moves through air. So this sudden explosive microbial respiration quickly drains every single molecule of available oxygen in the water column faster than it can be replenished.
It's essentially like a party where someone orders way too much food, and that's the runoff, basically. The guests eat themselves into a massive food coma, and in the process, they literally breathe up all the oxygen in the closed room until nobody else can survive.
That is a great analogy, a food coma where you drain the room of oxygen precisely. The invertebrates die. The fish suffocate.
And plants.
Plants can't grow because the initial massive bloom blocked all the sunlight anyway.
Yeah.
So the only organisms left surviving in that zone are anaerobic microbes that just don't rely on oxygen at all.
Wow. It's terrifying how quickly that scales up. But it also highlights something fundamental, I think. The chemistry of the water dictates who gets to live there. And the textbook draws a really sharp contrast between how freshwater and seawater function as habitats, specifically in how they handle carbon dioxide.
The difference is night and day. Let's look at freshwater first, like your typical lakes and streams. These systems are largely unbuffered.
Meaning what?
Meaning their pH is dictated by the dissolved carbon dioxide that sits in equilibrium with the air above it, which naturally makes freshwater slightly acidic, usually hovering around a pH of 5.0 to 5.5.
Okay, got it.
The pH in a river or lake is mostly at the mercy of whatever washes in from the surrounding land, whether that's acidic soil or alkaline minerals. And, how fast local photoautotrophs, like diatoms, can suck the CO2 out of the water for photosynthesis.
I always just assumed the ocean worked the same way, just, with salt. But the ocean has a totally different defense mechanism, right?
A massive one, yeah. Seawater is strongly buffered. It operates on this continuous chemical chain reaction. There's a great diagram in the book, figure 29.1, called the carbonate equilibrium system.
Right, which sounds super complicated.
It sounds dense, but think of it as a massive chemical shock absorber. When atmospheric CO2 dissolves into the seawater, some is used by marine life, sure, but a massive portion reacts directly with the water molecules to form carbonic acid.
Carbonic acid, okay.
But that carbonic acid is unstable, so it quickly dissociates, breaking apart into bicarbonate and carbonate. The relative concentration of that bicarbonate and carbonate shifting back and forth is what creates the buffer.
Oh, I see.
Yeah, it naturally stabilizes the ocean, keeping it alkaline, tightly locked between a pH of 7.6 and 8.2.
But wait, if the ocean has this massive built-in chemical thermostat, why is ocean acidification constantly in the news? It seems like it should be protected from that.
Well, it is protected, up to a point. The problem is that human activity has pumped out so much CO2 that the ocean is currently absorbing about 25% of all anthropogenically produced carbon.
Oh, wow.
Yeah. It is simply more carbon than the shock absorber can handle. We are overloading the system's capacity, and as a result, the average pH of the ocean has already dropped by 0.1 units since the pre-industrial age.
Which might sound like a tiny fraction to someone casually listening, but pH is a logarithmic scale. A 0.1 drop is a massive shift in acidity.
Massive.
Yeah.
And it fundamentally alters the physical building blocks of the ocean. The text highlights this in figure 29.2, showing a microbe called Amelia Huxley.
Amelia Huxley.
Right. It's a type of protist known as a coccolithophore. These single-celled organisms are incredibly important because they produce about a third of all the marine calcium carbonate in the ocean.
A third? That's huge.
It is. They use it to build these beautiful, intricate, plate-like armor shells around themselves. They're called coccoliths. But as the ocean's pH drops, the entire carbonate equilibrium shifts away from calcium carbonate. The chemical building blocks simply become scarce.
So by tweaking the pH, we are chemically altering a microbe's ability to build its own house.
And not just microbes. Larger organisms like corals rely on the exact same chemistry. So we see how the ocean protects itself chemically, but that raises a fascinating question.
What's that?
What happens when that massive, stable, buffered body of ocean water violently crashes into an unbuffered freshwater river?
The coastlines, estuaries.
Exactly. Estuaries are semi-enclosed coastal regions where rivers meet the sea, driven by tidal mixing. And from a microbial perspective, this is a daily torture chamber of extreme osmotic stress.
Because of the salt.
Right. With every single tide that rolls in and out, the salt concentration swings wildly.
I'd imagine standard freshwater microbes would just shrivel up and ocean microbes would burst. How does anything survive in that mixing zone?
Well, the resident microbes have to be halotolerant. That doesn't mean they require high salt to live, but rather that they can withstand the massive fluctuations.
Okay, so how do they do that?
They do this by manufacturing compatible solutes inside their cells. Fungi and protists might produce a compound called dimethyl sulfonyl propionate.
Say that 3 times fast.
I know, right? Bacteria and archaea might actively pump potassium into their cells or synthesize compounds like proline and betaine.
Okay, so they're basically packing their insides with these solids.
Yes. They perfectly balance their internal pressure against the wildly changing salt pressure of the water outside.
But coastlines are also where all our agricultural runoff hits the sea, bringing us back to that massive influx of nutrients we talked about. We discussed dead zones, but there's an arguably more immediate danger with coastal nutrient dumping. Harmful algal blooms, or HABs.
Yeah, eutrophication has a very dark side here. If nutrients are dumped into an estuary in a massive pulse, rather than like a steady trickle, it throws the balance off. A single photoautotrophic species can suddenly outcompete everything else and take over the water.
And some of these are toxic, right?
Incredibly dangerous. For example, there are diatoms in the genus Pseudonychia that regularly balloon off the California coast. They produce a potent neurotoxin called domoic acid.
And that scales up the food chain quickly. Like the anchovies eat the toxic diatoms, the toxin concentrates in their bodies, and then the sea lions eat the anchovies and die from neurotoxicity.
It cascades violently. Dinoflagellates are major culprits too. Certain species cause paralytic shellfish poisoning, while others, like Karenia brevis, cause the infamous red tides in Florida.
Right, the red tides.
Those blooms are notorious for producing toxins that literally become airborne in the sea spray, causing respiratory issues in humans on the beach and killing off endangered manatees in the water.
So coastal sediments are these incredibly complex, highly stressed, muddy layers of life, constantly shifting in salinity and oxygen. If you're a microbiologist, how do you even begin to study a muddy mess like that without going insane?
You build a model, specifically an elegant closed system model called a Winogradsky column, named after the microbiologist Krei Winogradsky. It's a brilliant way to visualize an entire salt-marred ecosystem in a single glass cylinder. You can see a diagram of this in figure 29.4.
Okay, paint a picture of this for me. What does it actually look like?
Okay, so you take a tall glass tube, fill the bottom with mud from an estuary, add some seawater, and mix in a source of organic carbon and sulfur.
What kind of carbon source?
Shredded newspaper works perfectly because it's packed with cellulose. You seal the top of the tube completely so no air gets in, and you place it by a window to give it light. Over several weeks, the mud separates into incredibly distinct, brightly colored microbial zones based entirely on chemical gradients.
Wait, but if it's completely sealed, how does it not just run out of oxygen and die in a few days?
Because the microbes build a self-sustaining engine. At the very top of the water column, where the light hits, cyanobacteria and diatoms start performing oxygenic photosynthesis. They produce the oxygen.
Oh, I see.
Just beneath them, aerobic heterotrophs consume that oxygen to break down the carbon. So the oxygen gradient is high at the top and completely fades away as you move down.
And what's happening at the dark, muddy bottom?
Total anoxia. At the very bottom, anaerobic fermenters, like Clostridium, are breaking down the newspaper cellulose. This feeds other bacteria like Desulfovibrio that perform anaerobic respiration using sulfate from the seawater.
Okay.
And as they breathe the sulfate, they produce hydrogen sulfide gas. That toxic gas naturally diffuses upward. So now you have oxygen diffusing down and hydrogen sulfide diffusing up.
And where those two invisible chemical gradients meet in the middle?
Well,
That has to be a hot spot.
it is the most fascinating part of the column. You get chemolithotrophs like Beggiatoa sitting right at the boundary. They use the toxic hydrogen sulfide welling up from below as their energy source, but they still use the faint traces of oxygen coming from above to breathe.
Wow.
And below them, where the oxygen is totally gone, but the light still penetrates the mud, you get vivid purple and olive green bands.
What are those?
These are anoxygenic photoautotrophs like chromatium and chlorobium. They use sunlight to fix carbon, but instead of using water like a plant, they use the hydrogen sulfide from the bottom as their electron source. It perfectly visualizes vertical carbon and sulfur cycling in a single tube.
A whole layered world running on waste gas and shredded newspaper. I love that. But if the estuary is this nutrient-rich mud bath, the open ocean is basically a desert. Moving away from the coast, how do microbes survive in the photic zone, that sunlit upper 100 meters of the open ocean where there is almost no available food?
They survive because they are forced to be the primary producers. Cyanobacteria, specifically species like Prochlorococcus and Sinetrococcus, perform roughly half of all the carbon fixation in the entirety of the open ocean.
Half. Just those bacteria.
Yeah. But to your point about the lack of nutrients, survival out there dictates a ruthless efficiency. Nothing can go to waste. The textbook maps this out as the microbial loop, which is shown in figure 29.5.
You know, whenever I think of the microbial loop, I picture an extreme zero-waste facility. Like if someone drops a piece of trash, someone else has to catch it before it hits the ground, or it sinks to the deep ocean, and those nutrients are lost forever.
That's a great way to look at it. The primary producers leak dissolved organic matter, or DOM, into the water as they grow. Viruses constantly burst open host cells, dumping huge amounts of organic matter into the sea. Heterotrophic microbes instantly swarm and consume this dissolved matter.
And then what?
Then, slightly larger protists, like flagellates, eat those microbes. The net effect is that essential nutrients, carbon, nitrate, phosphate, are fiercely, constantly recycled within that sunlit zone, actively preventing them from falling into the abyss.
But no system is 100% efficient. I mean, some of it has to fall.
It does. And the particulate matter that escapes the loop is known as marine snow, shown in figure 29.6. If you watch footage from deep-sea submersibles, it looks exactly like a snowstorm.
But it's not snow.
No, those flakes are actually flocculent clumps of fecal pellets, dead cell debris, and indigestible diatom shells drifting downward into the dark.
It's like a skyscraper being demolished midair. As the rubble falls, a crew of scavengers is frantically dismantling the concrete and steel while it's still dropping, so almost nothing actually hits the ground.
And those scavengers are microbes that rapidly colonize the falling snow. They form dense biofilms on the sinking particles, secreting specialized exoenzymes to break down the complex debris into usable energy as they plummet into the deep.
That's incredible.
But back up in the nutrient desert at the surface, these microbes have evolved some insane life hacks just to scrape by, particularly when it comes to energy and nitrogen.
Right, because nitrogen is crucial, but fixing it usually requires an oxygen-free environment. How do you do that in the sunlit, oxygen-rich surface?
You get incredibly creative. There's a marine cyanobacterium known as UCYNA. Most cyanobacteria fix nitrogen at night to avoid the oxygen they produce during photosynthesis. But UCYNA completely ditched its photosystem II machinery.
Wait, really?
Yeah, it literally evolved to stop producing oxygen entirely. By stripping away that machinery, it can comfortably fix nitrogen during the bright daytime.
That is a staggering evolutionary workaround. Just drop the oxygen production entirely. And there are other bacteria doing something similar just to harvest energy from the sun without doing full photosynthesis, right?
Yes, there is an incredibly abundant order of bacteria called Pelagibacteriales. They utilize a light-driven proton pump called proteorhodopsin. Right. They aren't doing the heavy lifting of full photosynthesis to fix carbon. Instead, they use the light energy directly to synthesize ATP. Other marine microbes use a pigment called bacterial chlorophyll to harvest light for ATP while getting their carbon organically. That's a process called photoheterotrophy.
Why not just use the organic carbon they find for energy?
Because organic carbon is incredibly rare in the open ocean. It is precious. By getting their daily operating energy, their ATP, directly from the sun, they can save whatever rare organic carbon they find strictly for building their cell walls and reproducing. They don't have to burn their building materials for fuel.
Okay, so we have this incredibly tight nutrient loop. We have microbes hacking the sun to save carbon and scavengers riding marine snow. But who controls the population? Who is the apex predator of the ocean? Because it's not a shark, it's a virus.
The numbers are almost incomprehensible. Viruses outnumber bacteria in the ocean 10 to 1.
10 to 1.
They are estimated to kill roughly 20% of the entire marine microbial biomass every single day.
If they are killing a fifth of their food source daily, how do they interact with host populations without just wiping everything out and starving themselves?
Microbiologists use three main models to explain this delicate balance. The first is the kill the winner model.
Kill the winner. Sounds intense.
It is. This suggests that as soon as one specific microbial strain becomes highly dominant and abundant, the winner of the ecosystem, it becomes the massive primary target for viruses. The virus crashes that specific population, making room for other, less dominant strains to thrive.
It's an automatic penalty for getting too successful. What are the other two models?
The piggyback the winner model relies on lysogeny. Instead of bursting the host cell, the virus inserts its own genome into the host's DNA.
Ah, so it hides.
Exactly. It just hides out and replicates peacefully right along with the host as the host thrives. Finally, there is the Red Queen model, which proposes a continuous, exhausting evolutionary arms race. The viruses and hosts are constantly mutating just to keep pace with each other, running as fast as they can just to stay in the same place.
And the visual proof of this viral power is immense. The textbook shows this in figures 29.10 and 29.11. It's a satellite photo of a bloom of the coccolithophore we talked about earlier, Emiliania huxley.
It is a stunning image. The bloom is 500 kilometers long. You can literally see it from space.
Wow.
And yet, its sudden rapid collapse is driven in large part by microscopic viral lysis. When viruses burst a bloom of that magnitude, they short-circuit the entire microbial loop. They instantly convert huge populations of living cells back into dissolved organic matter, or DOM, and particulate organic matter. They shatter the food chain and reset the nutrient pool.
It's a staggering scale. But eventually, everything that escapes the microbial loop, the marine snow, the viral debris, finally hits the absolute bottom, the deep benthos. And this is where the rules of biology just seem to break down entirely.
The benthos is an alien world. It's completely dark. The pressure is immense, meaning the microbes there have to be piezophilic. Right. And it's near freezing, requiring them to be psychrophilic. Because of the extreme cold and the absolute scarcity of nutrients in the deep abyss, their metabolic rates slow to a crawl. We are talking about microbial doubling times measured not in minutes or hours, but in hundreds or even thousands of years.
It's hard to even wrap your head around an organism taking a century to divide once. But if it's completely dark and buried in mud, what are they even breathing? How do they get energy?
This relies on the strict laws of thermodynamics. As you dig deeper into the sediment, oxygen disappears immediately. Microbes are forced to use different electron acceptors for anaerobic respiration, and thermodynamics dictates a very strict pecking order based on which chemical provides the highest energy yield.
Meaning they will always eat the easiest, most energy-rich chemical available first.
Precisely. Figure 29.1 maps out this upside-down energetics. Once oxygen is gone, the microbes use nitrate. When the nitrate is completely exhausted, they move to manganese, then iron, then sulfur. And finally, at the very bottom of the energy barrel, they use CO2, a process called methanogenesis. It is a predictable, deeply understood stratification of life in the mud.
So thermodynamics dictates this strict pecking order based on energy yield. Does it stay that way all the way down to the core of the Earth?
This is where the textbook throws a massive curveball.
Yeah.
It does not. The twist revealed in deep core samples taken off the coast of Peru is that once you drill deeper than 400 meters below the seafloor, this predictable profile suddenly reverses.
Reverses?
Yes. Methanogenesis and sulfate reduction start occurring at the exact same time. It completely baffles scientists.
Wait, if the rule is they always use the easiest energy first, a reversal means it's like finding a ball rolling up a hill.
It completely defies our standard thermodynamic models of the ocean floor. It points to mysterious, entirely unknown sources of electron acceptors welling up from deep within the Earth's crust. There is a massive energy engine driving life deep inside the planet that we literally cannot see and barely understand.
Upside-down energetics. That is mind-bending. But from the alien depths of the abyss, let's rise back up to the surface and move inland. We need to cover freshwater ecosystems, which is the tiny fraction of water that terrestrial life actually relies on.
Right. Ecologists divide these into lotic systems, which is flowing water like rivers and streams, and lentic systems, which are still waters like lakes. In flowing lotic systems, the microbial community can't just float or they'd wash away.
Right.
They are heavily dependent on creating dense, sticky, benthic biofilms attached to rocks and sediments.
And rivers are highly susceptible to sudden influxes of pollution. The textbook visualizes this in figure 29.16 with something called a dissolved oxygen sag curve. Imagine a graph tracking oxygen levels down a river.
Right, so imagine what happens when a single source of organic waste, say, a massive raw sewage, hits a clean river. Immediately downstream from the spill, there is a massive sudden dip or sag in the dissolved oxygen line on the graph.
Why does it sag?
This happens because heterotrophic microbes instantly gorge on the sudden influx of sewage waste. They respire rapidly, depleting the oxygen in that stretch of the river.
It's a localized, highly concentrated version of the ocean dead zone we started with.
But rivers flow. As you move further downstream, the waste is finally consumed and diluted. The oxygen slowly diffuses back into the water, and the phototrophic microbes reestablish themselves, allowing the oxygen levels on the graph to recover entirely.
Now, what about still water? Lentic systems like deep lakes behave very differently because they don't flow, they stratify.
Exactly. Figure 29.17 visualizes this beautifully. Imagine a deep lake in the dead of summer. It naturally forms distinct separated layers. The top layer is the epilimnion warm sunlit, packed with phototrophs.
Okay.
Below that is a sharp boundary of rapidly dropping temperature called the thermocline. And at the very bottom is the hypolimnion cold, dark, and frequently depleted of oxygen. These layers stay rigidly separated all summer.
But then the seasons change.
Right. When autumn arrives, the air cools. The surface water cools, becomes denser, and sinks. This causes the entire lake to physically mix, stirring up nutrient-rich water from the dark bottom and bringing it into the sunlit surface.
And that sudden mixing of nutrients frequently triggers MABA of microbial blooms. But when those blooms happen due to eutrophication, like from fertilizer runoff into a lake cyanobacteria, almost always seem to win the battle for dominance. Why are they so perfectly equipped to take over a polluted, high-phosphorus environment?
They have three distinct, brutal competitive advantages. First, they are incredibly efficient at grabbing trace amounts of CO2 from the water. As they frantically remove the CO2, they drastically raise the pH of the lake, making the water too toxic and alkaline for many competing protists to survive.
Wow, so they basically poison the competition.
Essentially, yeah. Second, many cyanobacteria secrete siderophores. These are compounds that act like biological chemical claws. They're released into the water to hoard all the available iron, physically starving out their competitors.
And the third advantage?
To make matters worse, some of these victorious cyanobacteria produce potent toxins and odor-causing compounds that can completely ruin a local drinking water supply.
Which brings us entirely full circle for you, the listener. We started with dead zones on the coast, but this microscopic warfare affects you everywhere. Whether you are dealing with a local toxic lake bloom, looking at sea foam blowing on the beach, or just taking a deep breath of oxygen, you are interacting with microbial ecosystems that are finely tuned to incredibly specific gradients of light, salt, and chemistry.
And it leaves us with an incredibly profound, slightly unsettling question to ponder. We just learned that the deep subsurface ocean contains a massive, unquantifiable portion of Earth's total biomass. These are microbes living on alien timescales of centuries, relying on an upside-down thermodynamic engine fueled by the Earth's crust itself.
Right.
So what happens is industrial deep-sea drilling and mining operations begin plunging into the benthos, churning up ecosystems that we barely even have the biological frameworks to understand yet.
If one pulse of fertilizer can suffocate a coastline, what does ripping up the century-old benthos do? It is definitely something to mull over. Thank you for joining us on this deep dive. Good luck on your microbiology exam. And from all of us here, a warm thank you from the Last Minute Lecture team. Keep questioning the unseen world around you.